Hi, and welcome back. Right now, we're going to be talking about biopsychology and behavior. So biology and behavior, or biopsychology. So I'm going to break this lecture into two parts because it's pretty long. And the first part that I'm going to talk about is going to be the brain. I want to start by showing you a video by Carl Schoonover. It's a TED Talk, How to Look Inside the Brain. The person presenting this is a neuroscience PhD student at the time that this video is made. And he takes us through understanding the brain and the advances that have been made. And he talks about the Golgi stain and how this particular thing moved the science forward because the stain was able to show us what was there. And this led to the modern visualization of the neuron. So this is a very important video for the topic that we're going to be going over. And the speaker talks about this in a very regular way. And it helps to allow us to understand the complexities of the brain and what it is inside of it. So I'm going to start by sharing my screen so that we can watch this video together. And here we go.
This is a 1,000-year-old drawing of the brain. It's a diagram of the visual system, and some things look very familiar today. There's two eyes at the bottom, optic nerve flowing out from the back. There's a very large nose that doesn't seem to be connected to anything in particular. And if we compare this to more recent representations of the visual system, you'll see that things have gotten substantially more complicated over the intervening 1,000 years. And that's because today we can see what's inside of the brain rather than just looking at its overall shape. Imagine you wanted to understand how a computer works, and all you could see was a keyboard, a mouse, a screen. You really would be kind of out of luck. You want to be able to open it up, crack it open, look at the wiring inside. And up until a little more than a century ago, nobody was able to do that with the brain. Nobody had had a glimpse of the brain's wiring. And that's because if you take a brain out of the skull and you cut a thin slice of it, put it under even a very powerful microscope, there's nothing there. It's gray, formless, there's no structure. It won't tell you anything. And this all changed in the late 19th century. Suddenly, new chemical stains for brain tissue were developed, and they gave us our first glimpses at brain wiring. The computer was cracked open. So what really launched modern neuroscience was a stain called the Golgi stain. And it works in a very particular way. Instead of staining all of the cells inside of the tissue, it somehow only stains about 1% of them. It clears the forest, reveals the trees inside. If everything had been labeled, nothing would have been visible. So somehow it shows what's there. Spanish neuroanatomist Santiago Ramon y Cajal, who's widely considered the father of modern neuroscience, applied this Golgi stain, which yields data that looks like this, and really gave us the modern notion of the nerve cell, the neuron. And if you're thinking of the brain as a computer, this is the transistor. And very quickly, Cajal realized that neurons don't operate alone, but rather make connections with others that form circuits, just like in a computer. Today, a century later, when researchers want to visualize neurons, they light them up from the inside rather than darkening them. And there are several ways of doing this, but one of the most popular ones involves green fluorescent protein. Now, green fluorescent protein, which oddly enough comes from a bioluminescent jellyfish, is very useful because if you can get the gene for green fluorescent protein and deliver it to a cell, that cell will glow green, or any of the many variants now of green fluorescent protein, you get a cell to glow many different colors. And so coming back to the brain, this is from a genetically engineered mouse called Brainbow, and it's so called, of course, because all of these neurons are glowing different colors. Now, sometimes neuroscientists need to identify individual molecular components of neurons, molecules, rather than the entire cell. And there are several ways of doing this, but one of the most popular ones involves using antibodies. And you're familiar, of course, with antibodies as the henchmen of the immune system. But it turns out that they're so useful to the immune system because they can recognize specific molecules, like, for example, the code protein of a virus that's invading the body. And researchers have used this fact in order to recognize specific molecules inside of the brain, recognize specific substructures of the cell, and identify them individually. And a lot of the images I've been showing you here are very beautiful, but they're also very powerful. They have great explanatory power. This, for example, is an antibody staining against serotonin transporters in a slice of mouse brain. And you've heard of serotonin, of course, in the context of diseases like depression and anxiety. You've heard of SSRIs, which are drugs that are used to treat these diseases. And in order to understand how serotonin works, it's critical to understand where the serotonin machinery is. And antibody stainings like this one can be used to understand that sort of question. I'd like to leave you with the following thought. Brain fluorescent protein and antibodies are both totally natural products at the get-go. They were evolved by nature in order to get a jellyfish to glow green for whatever reason, or in order to detect the coat protein of an invading virus, for example. And only much later did scientists come onto the scene and say, hey, these are tools, these are functions that we could use in our own research tool palette. And instead of applying feeble human minds to designing these tools from scratch, there were these ready-made solutions right out there in nature, developed and refined steadily for millions of years by the greatest engineer of all. Thank you.
Okay, so that was a video that I thought was pretty important to be able to watch so that we could take a look at brain and behavior, and start by taking a look at the brain. What we know about our brain is that our brain houses our thoughts, emotions, personality, and it orchestrates our behavior. It files our memories, and it's involved in our every move. It's the most complex component of the nervous system. The nervous system conveys messages throughout our bodies using electrical and chemical processes. And we'll be talking about the brain, spinal cord, and nerves. The brain has between 86 and 100 billion nerve cells. So for those of you who are interested in how your brain works, and for those of you who may be going on to work in fields in biopsychology or in medicine or any field involving the brain, there are some great TED Talks focused specifically on the brain. So I will make this link available to you. It'll be in the folder for this chapter in the course units. So if this is of interest to you, there is a whole list of talks that are geared just around how the brain works. So if we move on and we start to talk about neuroscience, this is the study of the brain and the nervous system. Biological psychology focuses on how the brain and other biological systems influence human behavior. Biological psychology is also known as biopsychology, psychobiology, neuropsychology, physiological psychology, and behavioral neuroscience. If you see any of those, they all really are the same thing. So where did all of this start? How did all of this begin? There is, I don't know how many of you have heard of the term phrenology. Phrenology is a now debunked science, but it was an early approach to explaining the brain functions by linking the physical structure of the skull with a variety of characteristics. So bumps on the skull, depressions on the skull, these were all thought to be meaningful. So if you were to go and touch your head is going to feel different than somebody else's head. It's a different shape, it's a different size. And so all of these bumps and differences and depressions were thought to be meaningful. Although this was found to be incorrect, it did advance the field and got people to start thinking about the brain having different areas responsible for different functions. So I just want to quickly share the screen again to show you an article by Britannica with an image of phrenology that they have there. So just a moment while I get that prepared. Okay, there we go. So I just want to show you this. I'm going to share my screen again. Okay, so what we have here is just an article on phrenology, but I want to show you the picture of what it looks like. So if you can see here on the skull, there it is portioned out, and in each area there is a, they believe that each of these areas signified a different function. So if we move down, there's a more colorful picture down here. So as you can see, they broke it off into different features that they believed had to do with a person's personality, memory, things like that. So this was our, although this was incorrect, this was the first idea of somebody saying, hey, I think that the brain may be involved in multiple functions. And so although this isn't something that we adhere to now, we do value the fact that this led to future work that we now rely on and that we now know does have meaning, and that is that the brain, different areas of the brain are responsible for different functions. So we didn't have a way to look inside the skull. So now we do, though. And we have many ways to look inside the skull. We don't have to rely on the outward appearance of the skull to look at the brain functioning that we have. So how can we do this? Well, I don't know if you've heard of an EEG, an electroencephalogram. This is where electrodes are placed on the scalp and they record electrical activity, and a computer printout shows the functional responses across the lobes. So let's take a look at what this looks like. Let's see. I'm going to just quickly show you a the output for EEG and the images that they show. So let me share my screen again with you so that you can see what I'm going to be looking at. So if you look here, I just put in EEG, and this is what it looks like. So a cap is placed on the skull, right? Like this, and there are electrodes. So let me see if you can see here. These are the electrodes. This would be a cap placed on somebody's head. Sometimes it's not a cap and somebody just places the electrodes right on the skull, like on this baby right here. So this is the idea that, and I'm scrolling down just to show you all of the different images associated with EEG. So here's what they were able to see. If you put the cap on and you have a person do a task, or you just want to measure what's going on in someone's brain, this is the output that you would get. This is what one looks like. It's these lines that are either long and flat, or they might be very high and low and close together. But this is how the printout looks. Now, this still doesn't show you what the brain looks like inside. It shows you what the electrical output is, but you still don't get to see what's really happening. And so this is kind of, this is a little tough in that respect, because you still don't actually get to see what the brain looks like. However, let's move on to the CAT scan, okay? Let's move to the CAT scan. So here are the images of a CAT scan. Many of you may have had a CAT scan. Perhaps you had a concussion or something like that and needed to go get a CAT scan. And it's not always of the brain. You can get a CAT scan of a different part of your body. But if we're talking about the head, this is what we're looking at. We're looking at a scan that looks like this, where you lay down and your head goes into this large donut-like opening here, and you need to keep your head very still. And this is what the scan itself will look like. Now, let me see if I can get the CAT scan images of the brain. So unlike the EEG that we saw, the output, which was lines. This right here is the output of a CAT scan. So these are actual images, and we're able to see inside the skull what's there. Now this is in black and white, and so we're able to see, you know, areas of the brain that, you know, where there are dark structures and things like this. This is a normal CAT scan of the brain. So this is what a normal one would look like. Let's see what one, this one, let's see if we can find one that has something. Let's see. Here's one where you can detect a stroke, right? So this darker image here. So these are the sorts of things that made a big change. We're able to now actually see the brain. So what else do we have to look at the brain? We also have an MRI scan. Some of you may have had an MRI, maybe, again, you had an injury. This looks very similar to the CAT scan. But in this case, in the case of a CAT scan, there's radiation involved. It's an X-ray. That's really what it is an X-ray. An MRI scan does not use X-ray. It does not use radiation. It uses magnets. It's magnetic resonance imaging. So in this case, we're able to see things a little more in detail. So look at this. This is different than what we were able to see with the CAT scan. So this whole imaging looks much different. And this is a plus. If any of you has ever gone into an MRI machine, the difference also between a CAT scan and an MRI is when you go into the MRI machine, you hear a very loud wah, wah, wah. They'll probably give you earplugs to put in, but it's very loud. And people who have claustrophobia often have a hard time with having your head inside of this machine. And you must hold very still also. So what's another thing that we have? We also have the PET scan. Now this also has radiation involved, where a radioactive substance is a dye is injected into the bloodstream, and you're able to get these really amazing images of the brain. So this is even more information that we're able to receive than from the MRI. So from here, you're able to see areas that are lit up. You can see different areas of the brain by the different colors that are showing here. So if you remember from the EEG, we see lines. The MRI is a black and white picture. The CAT scan is the black and white picture. The MRI gives a little bit more information. That PET scan really gives a nice colorful picture. And I'm also going to show you the fMRI. So this came out later. And in fact, this also gives really great colorful details. You're really able to see a lot. You're able to actually, they call it an fMRI machine because you're able to do functional activities while you're there. You don't have to remain still like you do with the regular MRI. And in this case, there's no radiation. Again, it's magnetic. It's the MRI, but it's called functional MRI, fMRI. So in this case, we still get the really amazing picture here, and we're able to see it in color, and we're able to get a lot of great information. So this is really how how the field has progressed, and being able to look at the brain with these variety of scans that are available, and this is how it has progressed. So these are really important to take note of, because this has helped to advance our field. So What we now know is, after looking at the brain and looking at all of the different uh, ways that we can look inside of our skull at our brain, let's move on to talk about what's going on inside the brain and go back to the video where I want to talk now about neurons. The neuron is the basic building block of the nervous system, and it has three parts: the cell body, the dendrites, and the axon. I'm going to teach you some really easy ways to remember this. Some of you may have already learned about this, but if you haven't, I have some really easy ways to remember this. So the cell body is the region of the neuron that includes structures that nourish the cell, and a nucleus containing DNA. It looks like a circle. So I want you to think of this fist that I have here. Make a fist for yourself. This fist is the cell body. Okay, think of it that way. The next thing I want you to learn about are dendrites. These extend from the cell body, and they're like little tiny branches or fibers that receive messages from other neurons and send information to the cell body. So these are dendrites. So you have your cell body, and attached to that cell body are your dendrites, okay? Next, we have the axon. And this is a skinny tube-like structure of a neuron that extends from the cell body, and it sends messages to other neurons from its terminal buds. So we have the cell body, the dendrites, and this is the axon. This right here, okay? Now, surrounding the axon is a myelin sheath. Many axons are covered in a fatty substance that insulates the axon and speeds the transmission of neural messages. So that myelin sheath, picture this would be like a bone underneath, that would be the axon, but the myelin sheath is the actual fatty covering of my arm. So that's just one way to imagine it. So right here, you have the way to remember this: the cell body, the dendrites, the axon, which is covered with a myelin sheath. Think of it like if you go down a slide in the middle of summer, it can be hot and bumpy, but if you add water to that, you slide down so quickly. So it's like a regular slide versus a water slide. That myelin sheath helps to make that message go much more quickly. So what we have also is a synapse, and that's the tiny gap between a terminal bud. I'm going to call my elbow the terminal bud. Okay, so if I'm receiving information in from my dendrite, it goes into the cell body, goes down the axon, and it has to come out somewhere. It comes out the terminal bud. Okay, so in between, there's a tiny gap between a terminal bud of one axon and the dendrite of a neighboring neuron. And this is where the communication occurs. So let me just demonstrate this. It's gonna be, just bear with me. It's not easy to do this with one person, but let's just say I have a message that comes in, okay? In comes a message. A message is coming in. It comes in, I pick it up from my dendrites. It goes into the cell body, down the axon to the terminal bud, and here waiting are the dendrites of another neuron. And in between is the synapse. See, it's not touching. I guess you can see it. It's not touching, but in between is the synapse, and the communication occurs there. The message is sent from this terminal bud right here through the synapse and into these dendrites, right? And now the dendrites take it in. It goes into the cell body, down the axon, out the terminal buds, and to the next set of dendrites. So it's like it's playing telephone in a way. Another thing to remember are glial cells. And these are cells that support, nourish, and protect the neurons. Some of them produce the myelin that's used on our axons. So glial cells are the glue that holds neurons together and maintains the structure of the nervous system. Okay, so glial cells are important. I want to share screen again with you to show you an image that I have that shows right here. This is what I was talking about. So although I use this, right, all of this as the image so that you can take this sort of cheat sheet along with you, you all have your own personal cheat sheet. So right here, dendrite. The nucleus, you'll have to imagine it inside of the cell body right here, but my fist is the cell body. Here's my axon with the myelin sheath on the outside of it. And you just have to picture it like a hairy elbow. And those would be the terminal buds that come off of it. Now, the direction of the electrical impulse always travels in the same direction. So it always comes in this way from the dendrite and goes on down. It doesn't then back up and go this way. It doesn't ever do that. It just goes in one direction, only in one direction. And down here will be another set of dendrites waiting for it. But dendrites don't pass anything on, they only take. So think of your hand motion like take. I'm taking, okay? So it only goes this way. So now I'm going to stop sharing here and continue on, but I want you to make sure that you remember some key things from this, and that is that the direction of the electrical impulse travels only in one direction. And remember, you're taking the information in with your hand like this, and it only goes in one direction. So I want to, oh, I had hoped that I had my phone with me. I'm going to pause this for a second, and I'm going to go get my phone, and I'll be right back. Okay, I found my phone. So I wanted to be able to show you this because when I have my phone here, I'm able to show you something kind of interesting that I think will be pretty helpful. So when we are talking about neurons and we're talking about how action potentials and those sorts of things happen, I want to first talk about this in terms of how it works with a phone. So there's something called resting potential. And this is when the electrical potential of the cell is at rest. The state of the cell when it's not activated. So right now I have my phone, and my phone is on, but it's not making any noise. It's not doing anything at all. It's just sitting here, but it's on. But if I didn't look at it, I wouldn't know anything about it. This is my phone in its resting potential. It's not ringing, there's no music on, there are no apps playing, nothing. It's just resting, but it's on. Now, an action potential is the spike in voltage that passes through the axon if a neuron, of a neuron. And the purpose is to convey information. So if I suddenly get a phone call, my phone will turn on, all right? So imagine, say, the cellular waves, the cellular or Wi-Fi are the dendrites, right? It's being picked up through the, I'm sorry, just imagine some dendrites here. There is a phone call that comes through over the Wi-Fi or the cellular, right? So it comes into the cell body. This would be the cell body. And it gets in and my phone says, ooh, there's a spike in voltage here. I know there's a phone call. I am going to activate, right? The phone call will go into the phone. It'll activate. It'll be just enough to go through. It'll go all the way from the dendrites into the cell body. It's no longer resting. There's a spike in voltage. It goes down the axon of the neuron, and this white part of my axon is covered in a white cord, right? That's my myelin sheath. And it goes all the way through into my terminal buds, right? That's what these are, my terminal buds. So I like to think of my phone, if I'm thinking of an action potential, as right now my phone is resting. It's on, but it's resting. But if a text comes through, or if a phone call comes through, something like that, that will be, we'll call that the action potential. It's a spike in voltage that passes through, goes into the axon, goes down the axon and into the terminal buds. So it's just like our hand, that we have this, it comes into our dendrites, goes into our cell body, down the axon, and out through the terminal buds. So I want you to think about this in terms of your phone, because we deal with this every day. We are dealing with action potentials and resting potential and all of this all the time. With our phones, there's an all or none property here, okay? And this is the same for a neuron. A neuron either fires or it doesn't. Action potentials are always the same strength. So how often have you been somewhere and you can't get a call because you're in the basement of a building or someplace that doesn't have reception? Somebody can be calling you, but it doesn't, the information doesn't get through to your phone, right? You can, so it's all or none. It fires or it doesn't. Another thing is that, you know, we talked about the myelin sheath and how it helps to get information through quickly. Have you ever had one of these cords where it's broken and part of the wiring shows through? And while you're talking, if it's not perfectly connected, you hear like, you know, it's hard to hear. Well, that's the same thing with our own neurons in our body as well. This myelin sheath is what protects everything and makes everything go smoothly. It works the same way with our phones and these cords here. So our terminal buds, it goes through the axon. It has to come out somewhere, and it comes out here, okay? These are our terminal buds. So think of your neurons in your body activating and running the same way that your phone does. So, and you can practice that with yourselves in terms of how the messaging goes. As you can see from the phone, it doesn't go from my ears, which are in the earbuds. My ears don't send any information in this direction. It only goes in one direction, comes into the phone and goes out through the terminal buds. So you can practice this on your own. Take a look at your own phone and remember that this is stuff that we see all the time. You know this principle, you understand how it works, and you actually carry around your cheat sheet with you at all times, which is you have your own dendrites, your own cell body, your own axon with the myelin sheath and terminal buds. And likely you have this as well. And so that can help you to remember how action potentials happen. So I'm gonna go give these earbuds back and I'm gonna come right back. Okay, so let's talk about the communication between neurons. We have neurotransmitters, and these are the chemical messengers that neurons use to communicate at the synapse. We also have receptor sites, and these are locations on the receiving neuron's dendrites where the neurotransmitters attach. Another term to know is the reuptake, and this is the process by which neurotransmitters are reabsorbed by the sending terminal bud. So I'm going to share my screen again, and I have a 2-minute neuroscience synaptic transmission video that's really a pretty good one that I want to be able to show you.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. The synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
Okay, so I think that that's a pretty good example of how it works. And so if you wanted to go back over that video again a couple of times, it's a good idea because it does show the process and how the synaptic transmission works. So let's move on to neurotransmitters and behaviors. There are many kinds of neurotransmitters, but here's just a few. I'm going to just talk about a couple of the few that are most commonly talked about. Acetylcholine. This neurotransmitter relays messages from neurons to muscles. It's also involved in memory, arousal, and attention. And dopamine is the coordination of muscle movement, attention, and pleasure. GABA inhibits communication between neurons. Glutamate promotes communication between neurons. And serotonin is involved in mood, appetite, aggression, and sleep. So let's talk a little bit about these. If we talk about norepinephrine, this prepares the body for stressful situations. It's involved in regulating sleep and arousal, and this helps the nervous system to initiate action to promote survival. Think of a soldier in battle. So in this case, if we are involved in a sort of a fight or flight situation where we're in a position where a bear is going to walk through your door and you think, oh my goodness, there's a bear there. We have our nervous system is initiating that action, which is going to promote survival by getting norepinephrine involved. Within that, serotonin is going to control our appetite, our mood and aggression, all of these things that we have going on. We also have dopamine, and this plays a key role in attention and learning through reinforcement and regulating body movements. And endorphins are naturally produced opioids. They block pain receptor sites, and exercise increases the production. So imagine yourself, a bear comes into the room and you say, oh my goodness, you're not gonna likely say, oh, how cute, I'm gonna go pet it. You're probably gonna think I need to get away. And when you do that, your neurons are gonna kick into action, and there will be messages that get sent to your muscles and to other parts of your body. You're not gonna be feeling hungry at this time, so your body is going to say, hey, don't focus on my appetite right now. Use all of my resources to send the send all of my resources to the muscles so I can run away and send this in make sure that all of the neurons know to send the information into specific places so my attention is focused on this, so that if I see something like a cute bunny over there, I don't say, oh, cute bunny. I am just focused on getting to where I need to go, and that's away from the bear. So all of these things sort of kick in and they work together, and this is something that happens within our body. So we have all kinds of neurotransmitters that are working, that have specific goals within our body, and they work to get us to do the things that we need to do. So let's now talk about the supporting systems of the hemispheres. We have the central nervous system, and this includes the brain and the spinal cord. So the way that I like to think of the central nervous system is just think of a lollipop, a big circle with a stick coming out of the bottom, okay? And then just put 2 eyes on it. That's your central nervous system. It's really easy. It's your brain and your spinal cord and, you know, your eyes. So think of it that way, and it's easy to always remember. We also have the peripheral nervous system, and this connects the central nervous system, right, the brain and the spinal cord, to the rest of the body. So think of that as all kinds of things coming out of the lollipop, okay? The brain and the spinal cord, so it's coming off of the spinal cord, all of these little like peripheral lines that are coming off of the spinal cord. So in that case, that's what the peripheral nervous system is. And the peripheral nervous system is made up of the somatic nervous system and the autonomic nervous system. So when we talk about the spinal cord, this is the bundle of neurons that allows communication between the brain and the peripheral nervous system. So the peripheral nervous system is connecting in here. I'm going to share my screen with you and show you the nervous system. This is an image of it. Right here, you can see the nervous system. So this right here is the central nervous system. This round here and straight line going down. The brain and the spinal cord is the central nervous system. The peripheral nervous system, as you can see, are these nerves that are connecting here. See all of these? These are part of the peripheral nervous system. So these, if you think of it in those terms, it's pretty easy to remember what's what. I have here let's see if I can pull this up, a video of the anatomy of the spinal cord and how it works. So let me see if I can get this and share my screen again so you can see this.
The spinal cord is a very important part of our central nervous system, the overall system that controls and coordinates the activities of our bodies.
Our brain is surrounded by the skull, while our spinal cord is surrounded by rings of bone called vertebrae, and both are covered by a protective membrane. Together, the vertebrae and membrane make up our spinal column, or our backbones. The backbone, which protects our spinal cord, starts at the base of our skull and ends just above our hips. The spinal cord is about 18 inches long and extends from the base of the brain down the middle of the back to just below the last rib in the waist area. The main job of the spinal cord is to be the communication system between the brain and the body by carrying messages that allow us to move and feel sensation. Our spinal nerve cells, called neurons, carry the messages to and from the spinal cord. These messages leave the spinal cord through openings in the vertebrae. Spinal nerve roots branch off the spinal cord in pairs, one going to each side of the body. Each nerve has a specific job for movement and feeling. They tell the muscles in your arms, hands, fingers, legs, toes, chest, and other parts of your body when and how to move. When a spinal cord injury occurs, sensation and movement may be interrupted, resulting in a temporary or permanent loss of function and paralysis. In general, The higher on the spinal cord the injury occurs, the greater loss of function the person may experience. The vertebrae are grouped into sections according to their location. For example, the 7 vertebrae in the neck are called the cervical vertebrae. The vertebrae are numbered top to bottom from the head to the tailbone. The top vertebrae is called C1, the next is C2, C3, and so on. Cervical spinal cord injuries usually cause loss of function in the arms and legs, resulting in tetraplegia, which is also called quadriplegia. The next 12 vertebrae are called the thoracic vertebrae. The first, T1, is where the top rib attaches. Injuries in the thoracic region usually affect the trunk and legs and result in paraplegia. The lumbar vertebrae are the 5 vertebrae in the lower back between the thoracic vertebrae, where the ribs attach, and the pelvis, or hip bone. The sacral vertebrae run from the pelvis to the end of the spinal column. Injuries to the 5 lumbar vertebrae, L1 through L5, and similarly to the 5 sacral vertebrae, S1 through S5, generally result in some loss of functioning in the hips and legs.
Whether the cause of a spinal injury or illness is traumatic or non-traumatic, the damage affects the nerve fibers passing through the injured area. That can damage or weaken part or all of the corresponding muscles and nerves below the injury site. Doctors have already or will assign your loved one a level of injury that corresponds to the damage below the injury. In Chapter 5, we'll explain more about these injury levels and how they can impact you or your loved one. Right now, we'll move on to Chapter 3 to inform you about some things that may occur soon.
Okay, so this is helpful because it shows how all of this works together and how an injury can actually cause problems here and how and why that can occur. So I'd like to talk now about some examples of some of the things that we test for and take a look at in the real world. Some things that are used. So we have all this information, right? We're able to test this, and why do we do it, and where is it important, and how does this impact anything in the real world? Well, let's take a look at one thing that is used pretty frequently. It's a real example of a test that's used in medical and psychological testing to screen for dementia. And when I say that it's used to screen for it, screening tools are never diagnostic. This is never something that is used to say, oh, here's a problem. It's just something to say, oh, if a person scores high on this, it's something, or low, depending on how the scoring is, that you would say, this is something that I want to look at further, and I want to make sure that I am going to look to see if this person needs further testing. So I'm going to share my screen, and I'm going to show you the MiniMental status exam. This is used all the time in doctor offices and in other settings by professionals to screen for dementia or brain functioning. So here we go. I'm going to see if this link works. Hopefully, yes, it does. Good. So this is the MiniMental state examination, and I put up a picture of what it looks like in this video because I think that it is important that you can take a look at it and see what's really used in the real world setting and why. And so learning about all of this brain functioning and how it works is very important for all of us. What is done here is each of these questions scores a series of points for each correct response. So in terms of the first one, what is the year, season, date, day, and month? The maximum score that you can get for that is 5. If they got one of those wrong, it would be a 4. 2 of those wrong would be a 3. That's where you would put the patient's score in here. The next question, where are we now? The state, county, town or city, hospital, or if you're giving it to in a doctor office, and what floor are we on? All of those could result in a maximum score of 5. In this next question, the examiner names 3 unrelated objects clearly and slowly, and then the instructor asks the patient to name all 3 of them. The patient's response is used for scoring, and the examiner repeats them until the patient learns all of them if possible. So I'm not going to go through all of these. If we were in a live classroom setting, I do go through this with the entire class and have everyone pair up. But because we're doing this digitally, I will not do that, but I will leave this link in this lecture folder so that you can go back, print it out, pair up with someone, and give it a try. The one thing to take note of is that it isn't just like you ask these questions willy-nilly of someone. There is a structured way to do it. And I do have in our notes a short video that I'd like to show you, because this does correspond with how you should do it. This is a student giving this. So go ahead and let's take a look at it together.
This is the short portable mental status questionnaire. And I'm just going to ask you a few questions, and I just need you to answer them as best as you can. All right. So the first question is, what is the date, month, and year?
What is the date, month, and year?
Yes.
Today's date?
Yes.
Tell you the month and the year. I don't know what to tell you.
Okay, that's fine.
Okay. Today is the day is Saturday. The month is July, and the year is 12.
Okay. What is the day of the week? Which you just said already. Can you say it again?
Saturday.
Thanks. Okay. What is the name of this place that you're at right now?
It's at home.
All right. What is your home phone number?
942-224-6207.
Okay. How old are you?
And I said that wrong. 972.
Oh, okay.
224-6207.
Okay, got it. How old are you?
I am 80. Yeah, I'm sorry.
Trying to trick us.
I'm sorry. I am 78 years old.
Okay. And when were you born?
Paris, Texas.
When? Sorry.
8/8/33.
Oh, okay, so 1933.
The 8th of August, 1933.
Okay. Okay. Who is the current president?
Barack Obama.
And who was the president before him?
George Bush.
All right. What was your mother's maiden name?
Coleman.
Coleman. Okay. Can you count backwards from 20 by threes?
From 20 by threes? No.
Count down backwards from 20 by 3.
You can't do it.
You can't do it?
No.
You don't want to
Let's
try?
start with 21.
Well, you can count. You don't have to get to 3. Just count backwards by 3. Can you do that?
About 3 hours.
From 20?
I imagine I can.
All right.
Okay. 18. Because I got to count 21.
Oh.
18. 15. 12. 9. 6. 3. That's it.
All right. All right, then.
Okay, so I just wanted you to get an idea. And I like to show that video because it's a student, the person is learning how to do it. So you can see that it's not exactly easy yet for her to do it. It's all of these always look much easier than they are, and you'll see if you go to try to learn how to do it yourself that it isn't as easy as it looks. But it is important to get to know how these work because these are the reasons we're learning about brain and behavior, and this is the way that biopsychology is useful to us. So Again, I'm going to leave these links for you to have so you can practice this on your own should you want to. And I will leave the link up also for the video as well. So I'm going to stop this lecture here because this is a long lecture today, and I will resume later, but this is the end of part 1 for today. So I'll see you in a little while.